So yeah, let's change gears a little bit. I'm gonna drive you into a more biology applied journey for today. And I will be telling you about how we investigate mitochondrial functions and how they're in interplay with the cell cycle by using a lot of sensors, and how we infer information from these sensors, so how we obtain information from the sensors to infer signaling regulation pathways. So if you're in the mitochondrial field, or even if you're not, you might recall this from your previous lectures at university, they are key organelles for cell homeostasis. So they are, first of all, the main producers of ATP for all sorts of metabolic reactions. They can act, of course, as signaling platforms, and their function in metabolism and in signal transduction is key to the development of tissues and organs. But they also counteract the production of reactive oxygen species that can act as a source of stress. And if the stress is too extended, they can also trigger programs of cell death. And you might have heard that mitochondrial dysfunctions are actually very widespread in a variety of multifaceted diseases. But from the signaling platforms, they are also a huge highway. And here it's showcased actually what's happening inside, or actually even a small fraction of what is happening inside the organelles. But what you should keep in mind is that they are not only an internal highway signaling to themselves, but they are also signaling outside of the cell. So they are in a constant interplay with the rest of cellular compartments. And they receive, decode, and produce output signals. And of course, from a signaling point of view, this is a source of, I would say, complexity in trying to decipher what is happening here and how mitochondria signal back. And in the lab, we're trying to dissect a small proportion of this complexity by trying to understand how mitochondria interact with the cell cycle machinery. And we do so by developing several approaches. For instance, first of all, we want to sense mitochondrial activities that oscillate throughout the cell cycle. And we do so, and you will see during my presentation, we do so by implementing FRET sensors. But we also aim to identify cell cycle proteins that act on mitochondria. And last, of course, we're signaling people, so we want to understand the signaling choreography behind this interplay, and this across 3 resolution scales. So from individual mitochondria up to the tissue level. And in today's presentation, we will see a little bit of all of this. First of all, I'm going to be telling you a story of how we detected a cell cycle protein which acts on mitochondria. So this protein is called Aurora A. And to do so, to uncover the roles of this protein on mitochondria, we mixtured FRET, biosensors, and also photoconversion in cells and in vivo. And also, I will be telling you about how, from these first piece of data, we engineered a novel FRET sensor to monitor intracellular turnover. So basically, a sensor to detect how the cell recycles its very own material through the autophagy pathway. So if you're in the cancer field, or if you're in a cancer institute, for instance, you might have heard that Aurora A is an important protein in cancer therapies. Why? Because it is actually a bona fide hallmark of epithelial cancers, so breast, ovarian, pancreatic cancers, for instance. And it's actually an old client since decades, because we know that the overexpression of Aurora A increases the size of the mammary gland, and you can appreciate it here in this histology section compared to a control one. But not only this increase in the size of the mammary gland leads actually to tumor formation, both in mice and in humans, with very poor survival, actually. And of course, if you have such good in a sort of a way, client, you want to have a look at the molecular activities of this protein. So we know that Aurora A is a kinase. It is a serine threonine kinase that actually is playing lots of roles in the cell. It has non-mitotic functions, and these functions take place throughout all the phases of the interface, so from G0 to S, going from DNA replication to neurite extension to transcription activation, for instance. But Aurora A is mainly known to be a mitotic protein. So its abundance, so its levels peak at the G2M phase of the cell cycle. And throughout mitosis, it actually orchestrates centrosome maturation here. But also it's acting as a timer for the mitotic phase. And last, it controls the construction and the stability of the bipolar spindle. So the structure that actually drives the separation of the chromosomes in the two daughter cells. But so far, I haven't mentioned mitochondria. So how did we link mitochondria with Aurora A? So actually, the evidence was pretty scarce when we started this project. So we know that epithelial cancers, so as I was saying, breast, ovarian, and pancreatic cancers, they rely a lot on mitochondria for ATP supply. So they are not glycolytic. They don't use the glycolysis for ATP. They use these tiny little things. And we know that in these very same cancers, Aurora A is hugely overexpressed. Of course, this was rather indirect evidence, but a seminal paper came out in 2012, at least indirectly linking Aurora A with mitochondrial segregation and mitosis. So with the capacity of the mitochondrial pool to be partitioned into the two daughter cells. And Aurora A does so, as I was saying, indirectly, because actually it's helping the relocalization of two substrates, RAL-A and RAL-BP1, at mitochondria. You might say, okay, but actually, we are signaling people, we are microscopy people, and I was really frustrated to, I was really interested in trying to understand how this relocation is happening, both in space and time. So basically, how is Aurora A mechanistically doing that? And if you believe to a certain extent, to the principle of Occam's razor, so that the simplest explanation is sometimes the correct one, we started to hypothesize that maybe Aurora A is doing this function at mitochondria because it is there, because we can find it there. And so narrowing down the distance between something that is potentially somewhere else in the cell, but bringing it close to mitochondria. So the first question that we asked was, okay, is it a mitochondrial protein? And I promise this is the only Western blot I'm going to be showing you today, but sometimes you're obliged to do them. So if you're in the mitochondria field, there is a classical assay that you do to establish if a protein is in mitochondria or not. And these assays are called trypsin digestion assays. So basically, if you're not familiar with those, you take your mitochondria from cells, you extract the mitochondrial population, and you treat it with trypsin to get rid of the outer mitochondrial membrane first. Or you treat them with trypsin and detergents to get access to the inner mitochondrial membrane. So here, there's invaginations of the inner mitochondrial membrane. And basically, you compare, this is how it looks like, only blot, as I was saying. You compare the degradation pattern, the digestion pattern of your favorite protein, so in our case it was Aurora A, with the digestion patterns of proteins that are known to be at mitochondria. So for instance, we selected TOM70 for the outer mitochondrial membrane, cytochrome C for the intermembrane space, and PMPCB for the matrix side, so the innermost side. And so you treat your mitochondrial fractions. Here you have the total ones for control, but you treat your mitochondrial fractions either with trypsin for increasing amounts of time alone, or with detergents, so digitonin or triton. And you just compare. And if you compare the degradation, so the digestion patterns, you will appreciate that Aurora A is getting digested very, very late. So you need trypsin and you need detergents to get rid of this protein from mitochondria. And this is a very similar pattern compared to the one of PMPCB. So Aurora A is getting digested late as a matrix protein is doing. So we were very happy with this because not only it is a mitochondrial protein, but it's imported and integrated into mitochondria as mitochondrial matrix protein do. But of course, this is just a localization. We started to wonder, okay, is it playing any role there? And since it is a kinase, of course, the first thing you would look at is, okay, is it active at this compartment? And to answer this question, we took advantage of our favorite technique in the team, which is FRED by FLIM microscopy. And so this is just going to be a brief reminder because Joachim and others very nicely introduced this. So in FRED by FLIM, you rely on an intrinsic property of the fluorescence, which is the lifetime, which is the time that it takes for one molecule of any fluorophore, in our case a donor, to go back to its ground state. And this is like a fingerprint. So it's a very, very stable property of fluorescence that actually can change its behavior when a donor and an acceptor fluorophore, such as GFP and M-cherry here, are in close proximity, so less than 10 nanometers. In this case, an excitation of the donor will lead to an energy transfer towards the acceptor, and in terms of lifetime, you will observe a shortening of the lifetime. So compared to a condition where you have no interaction, if you have a protein-protein interaction, you would see a decrease in the lifetime. Okay, but how do we apply this to Aurora A and to answer whether it is a mitochondrial active kinase or not? Well, we created what we can consider a biosensor. So we actually performed FRET with just one protein. We took the full-length sequence of Aurora A here, and we flanked it with a donor and an acceptor fluorophore. So in this configuration is GFP and M-cherry. And we benefited from data coming from the crystallography field, where we know that that Aurora A has at least two conformations, an open, inactive one here, and a close, activated one here. This change in conformation is induced by an autophosphorylation of the kinase, so the kinase can autophosphorylate, auto-modify itself on this residue. And this actually leads to a closure of the kinetic pocket, which also brings the two termini together and allowing, therefore, the FRET effect. So whenever you see a conformational change, so whenever you see FRET, you can infer on the conformational change related to the activation of Aurora A. This sensor, so this is the first version of the sensor, was particularly instrumental to reveal novel functions of Aurora A. So we expressed our sensor at endogenous-like levels, and we started to monitor whether Aurora A could be active only at mitosis or in other cell cycle phases. And this was the first piece of evidence that confirmed an activation of Aurora A at mitosis. You see here the poles of a mitotic spindle. Aurora A is located there. This is a fluorescent image of our sensor. And here you see a lifetime cartography, where you go from high lifetime values to low lifetime values. And so you already see in this map here, in this pseudo-color scale, that the lifetime of the Aurora A sensor at mitosis is pretty low. It's towards the yellow pseudo-color scale levels. And you can see it here showcased in the graph. So if you compare the behavior of the sensor, the behavior of the biosensor here in white at mitosis, you will see that it is statistically different from a donor-only construct. So a GFP Aurora A that carries no acceptor and therefore is incapable of performing FRET. But to our great surprise, actually, the expression of the sensor in other cell cycle phases led us to observe that there is a strong activation of the sensor at centrosomes during G1. You can see it here. And this activation was completely undiscovered before, was completely unknown, and it's actually as strong as the activation of the sensor in mitosis. Very interestingly also, if you look at centrosomes, so FRET measurements were done on these very bright spots, which are the centrosomes. If you look at cells in S phase, you will see that there is no activation, of Aurora A at this location, which results in non-significant changes in the lifetime between the biosensor here in white and the donor only. Okay, so we said if there is an activation of the kinase, there should be, or there could be, a potential role of the kinase at this compartment. And actually a new role for Aurora A at this, during the G1 phase was detected concerning microtubule elongation, because if you treat the cell with a thermic shock and a depolymerizing agent called nocodazole to take out the microtubules from the cell, and then you wash out the insult and you look at microtubules regrowing progressively over time, you will see that in control cells that express alpha-tubulin, GFP, microtubules will nicely regrow and go towards the edges of the cells over time. But in Aurora A depleted cells, this regrowth process cannot occur. And microtubules are actually very fragmented and actually stop there. Okay, so this was a first piece of evidence for the usefulness of the sensor concerning Aurora A, but we also identified few limitations of our tool. First of all, The delta lifetime, so the net difference in lifetime between the donor-only construct and the biosensor in our hands was maximum 120 picoseconds, which is okay, but it's not huge. And the GFP M-Cherry configuration actually is preventing us from testing the Aurora A sensor with another sensor, so a process called multiplex. Because Aurora A is a kinase, so you want to see its activation, but maybe you also want to see its activity towards the substrates because it's phosphorylating substrates. So if you want to use multiplex FRET, this configuration might be not super useful due to the spectral properties of the sensor itself. And on the biological side, there is also another problem, is that FRET by FLIM experiments actually require adequate protein expression levels because you need a certain photon budget to perform the experiments. And this is particularly not convenient for Aurora A. Why? Because actually its levels are different according to the cell cycle phase. So in G2M we have a lot of Aurora A, so it's easy to perform the measurement, but in G1 they are rather low. And this is also particularly problematic if you look at specific subcellular compartments. That's why basically so far I showed you only FRET on centrosomes or on the mitotic spindle, because it's actually a very bright spot and you can detect it regardless of the cell cycle phase. But if you want to perform FRET in the cytosol, for instance, in G1, the kinase is so, we have such a poor abundance, such a low abundance of the protein that is actually hard to perform FRET by FLIM. That's why we started to implement or upgrade the biosensor by producing novel versions. And so there were two major update routes that we wanted to take. The first one was to implement multiplex FRET approaches. So you will see data concerning fluorescence anisotropy that we use to potentially detect HOMO-FRET, and dark acceptors, again, that allow us to accumulate our first sensor with the second one. And secondly, we also wanted to have a better dynamic range from our sensors. So we also tested novel donor acceptor pair to improve the delta lifetime differences. So first thing first, we started with fluorescence anisotropy. So we had a GFP M-Cherry version of the sensor. We said, okay, what if we replace the acceptor with another GFP? So we have a GFP Aurora A GFP sensor. This is particularly convenient because we know that HOMO-FRET can be detected with anisotropy, and that anisotropy decreases in the presence of FRET. And so basically, you illuminate your sample with two laser excitations, a parallel and a perpendicular one, and you calculate an anisotropy index. And as you can see here in this picture, so we compared GFP Aurora A, so the donor alone again, the biosensor GFP Aurora A GFP, and from now on you will always see one mutant that we constantly use in our analysis as the lysine 162 methionine, which is an inactive mutant of Aurora A, keeping the kinase open like this. So for us it's a very important biological control. And as you can see here in the micrograph, so if you focus on these anisotropy pseudo-colored images, or also if you want to take a look at the graph, well, we were very disappointed because we couldn't see any FRET occurring. So any loss in anisotropy corresponding to FRET in any of the conditions we were testing. So we were very sorry, but we couldn't detect any FRET using this tool. So this is why actually we felt obliged to take a secondary route, an alternative route, and we focused on dark acceptors. So this was an exciting time where dark acceptors, so non-fluorescent proteins such as shadow G, for instance, were starting to be engineered, and they were showed to be promising because not only they were non-fluorescent, but they were also behaving as excellent acceptors for donor proteins. So shadow G was the first one that came out historically, and it was optimized for cyan donors such as M-turquoise2. And so here we basically took cells in mitosis, so where we knew that Aurora A was abundant. And you will see here mitotic spindles from now on. So here, for instance, and here, and you can compare the donor only. So this is always our standard control. So an Aurora A M-turquoise2 with or without shadow G. And as you can see here, we start seeing decreases, we start to see differences in the delta lifetime profile. profiles, which are also showcased here in the graph. So the delta lifetime difference, so the FRET occurring between a donor-only condition and a biosensor, is significant here. And with an inactive mutant, we lose at least partially the FRET effect. So we were very pleased with that, because actually now we have the possibility of accumulating two sensors at the same time, and this will come up later in my presentation also. But we also noticed that we had a difference, a net difference between the donor only and the biosensor condition, which was roughly 150 picoseconds. So slightly better than our GFP cherry one, which was 120, but nothing superior still. So this is why we, at the same time, we were focusing on improving our sensor and the dynamic range of it by replacing the donor and the acceptor pair. And so we were very grateful to Marie-Aurore and her team that provided us with a new bright yellow fluorophore, which is called nowadays TD-LAN YFP. Back then we were calling it Super YFP, so in the original publication you will see it quoted as Super YFP. But we put it in a donor acceptor pair with M turquoise 2, so the very same fluorophore as before, being a donor of energy. And again, the principle is always the same. You compare a donor-only condition where Aurora A is expressed at mitosis on the mitotic spindle, and then you compare it with the biosensor, so carrying Super YFP. And as you can see here, not only in these pseudo-colored images, but also on the graph here, the difference between a donor-only condition and the biosensor dramatically increased. So we still have a partial loss of the lifetime with an inactive mutant, but also we saw that the difference between these two conditions here is also, it can go up to 250 picoseconds. So we nearly doubled the delta lifetime difference compared to the first version of the sensor. And you might ask, why are you interested in doing so? Well, because maybe we reasoned that with this type of tool, we could finally go in the cytosol of G1 cells and measure FRET. So we, since the expression levels of Aurora A are so low in the cell cycle phase and at this compartment, we implemented more sensitive methods to sense FRET in the cytosol, and these methods rely on two-color FCCS. And again, you will compare. We compared the profile. I mean, the autocorrelation curves from the wild-type sensor here and the cross-correlation curve here for this construct, compared to an inactive version of the sensor. And as you can see, there is a dramatic change in the ratio between the cross-correlation and the cyan autocorrelation, which is lower, thereby indicating FRET with the wild-type sensor, and which is higher, indicating a loss of FRET with the kinase dead sensor. So again, accumulation of approaches and of new donor acceptor pair to ameliorate our FRET readout, and to potentially to detect pools of the proteins which were undetectable before. And right now, we clearly have a palette of Aurora A sensor for biology, so relying not only on standard FRET by FLIM, but that can be implemented for multiplex FRET, and that can be more sensitive, for instance, allowing us to use two-color FCCS. And now, we can finally go back to mitochondria, right? Because these are great tools for us to go and sense Aurora A pools in discrete subcellular locations. Nevertheless, the first experiments that we performed were done with the version 1.0 of the sensor, so GFP cherry. And as you can see here, again, the experiment is standard in our hands. We compare the donor-only condition here, and by expressing endogenous-like levels of the kinase, we see that we can detect a discrete pattern which corresponds to the mitochondrial pattern here, which with its own lifetime. And this lifetime decreases when we have a biosensor construct. So GFP Aurora A cherry. No news under the sun. You've been seeing this before in my previous slides. It's just that this time it is at mitochondria, and very interestingly, this pool is sensitive to inhibitors. So if we take inhibitors that actually are used in the clinic to counteract Aurora A specific cancers, we can actually revert this activation of the sensor. So meaning that there is no more difference between a donor-only condition and the biosensor condition. These compounds derive from MLN type of compounds, and this compound is the most promising one that we have, although it has a poor efficiency in vivo, but it's called MLN 8237. So We retrieved an activation of the sensor at mitochondria. What for? And of course, if you're in the mitochondrial field, you're going to be interested in looking at the mitochondrial morphology. And so we performed these experiments in cells, and we benefited from the collaboration with Thomas Rival in Marseille that actually provided us with a very nice model of Drosophila expressing Mitochondria 2. So Mitochondria 2, I mean, it's a photoconvertible protein. You heard this before during the week so far, but just a brief reminder. So it is expressed only at mitochondria. And when you photoconvert a subset of mitochondria in the row, you can have two outputs. So either they are not connected. So upon photoconversion, mitochondria will become red, but over time, mitochondria will remain in the region of interest or in the close vicinity. Or your mitochondria might be connected, and you're going to be experiencing the effect of water in a pipe. Basically, it will diffuse through the matrix, and you will reach mitochondria or pieces or portions of the network which are more far away. And so this is exactly what we did in Drosophila. In Drosophila, we have a very nice model to keep the fly alive. And to image in a non-invasive manner mitochondrial functions in the notum, so which is the back of the fly itself. And here you see an epidermal cell of the notum of the fly expressing Mitochondria 2. This is before photoconversion, so everything is green and no red. And you see your tiny little mitochondria. Some of them are behaving as threads. Some of them, they are dots. And after photoconversion, so we photoconverted in this ROI here, our mitochondria become red, but over time they are mainly staying in the ROI or in close proximity of it, indicating that the connectivity in this case is limited. But if we overexpress Aurora A, here again it's before the photoconversion, so this is a cancer-like paradigm. And if we photoconvert in this ROI, you will see that over time, almost the entire mitochondrial network becomes red. So meaning that mitochondria is diffusing throughout the network. And actually, this is happening not only at 2 minutes, this is happening very, very rapidly, meaning that the pipe is really working and water is like really flowing. And if we quantify mitochondrial connectivity by normalizing mitochondria red objects at different time points, we can actually see that we have a higher connectivity here in flies that overexpress Aurora A compared to wild-type flies. So, long story short, you overexpress Aurora A, like in cancer, and you induce mitochondrial hyperfusion. Is this just a cosmetic change of your mitochondrial shape? Well, the answer to this question is no, because again, if you're in the mitochondrial field, you might be wanting to know how much ATP your mitochondria are producing. So one way of assessing ATP levels is through mitochondrial respirometry assays. And so we provided evidence that this conformation of mitochondria actually is increasing mitochondrial respiration. So mitochondria produce more ATP when Aurora A is overexpressed, compared to cells, for instance, that express only a GFP. So we have a selective advantage for these cells. They change the mitochondrial shape in an Aurora A-dependent manner, and this induces more ATP in the cell. So to cut a long story short, so far I showed you that Aurora A is the mitochondrial protein, is getting inside the organelles, it's reaching the matrix. And the fact that you have a cancer-like situation where you have an overexpression of Aurora A leads you to have a hyperfusion of organelles. So you have this gigantic mitochondria. They are not just gigantic, but also that produce a lot of ATP. And actually, if you're a cell biologist like me, you want to know how this is happening, right? So you want to start looking for the partners in crime for these roles of Aurora A. So we started looking for partners, and we were benefiting, first of all, from our own proteome of Aurora A, which was making actually life harder because we were detecting approximately 250 partners of Aurora A at mitochondria, which, yeah, it's a little bit complex to dissect. But actually, we were helped by the literature because a seminal paper came out depicting one key partner of Aurora A, which was prohibitin 2, that in this study was found to be the receptor of LC3 on the inner mitochondrial membrane. And if this looks like Chinese to you, don't worry, I'll guide you through. Why is this important to us? Well, Again, if you're in the cell biology field, you might have heard of a pathway to clear the damaged material from the cell, which is called autophagy. And LC3, so this autophagy pathway actually consists in building double membrane vesicles called autophagosomes that actually grow and grow and grow and encapsulate the intracellular material, including mitochondria. And LC3 is a key partner in this pathway. So without LC3, it's really, really hard to do autophagy. And LC3 is also very peculiar because it's there from the beginning to the end of the pathway. So at the end of the pathway, the autophagosome here will fuse with the lysosome, and thanks to the acidic pH of the lysosome, you will degrade the cargo in the vesicle itself. And so we went back to our FRED by FLIM analysis. We were very intrigued by all these potential partners of Aurora A and mitochondria. And first of all, we validated our hit that we obtained from the interactomic analysis by performing FRED by FLIM in a standard protein-protein interaction mode. And as you may envisage here, we have Aurora A GFP, which is the donor. And again, here you have the donor itself. And we have this time two acceptors because prohibitin 2 is found in a complex with its cognate protein prohibitin. And we tested their mitochondrial. And so we tested FRET at the mitochondrial, on the mitochondrial surface here, which is dashed in black, concerning prohibitin and concerning prohibitin 2. And you can already foresee that we have significant changes in the lifetime only in this mitochondrial area. And for your convenience, this is quantified here. And we detected a significant FRET between Aurora A and prohibitin, but FRET was even more significant between Aurora A and prohibitin 2. And at this point, if you remember what I just told you, meaning that there is a third protein in this game, we started wondering whether it takes only 2 to tango, or maybe sometimes it takes also 3 to tango. So we started to probe protein-protein interactions 2 by 2 using FRED by FLIM. This is what I just showed you before. So Aurora A, it interacts with prohibitin 2. But also we found out that it interacts with LC3 itself. So the second protein is still effectively in the game. And this is dynamic. If we remove prohibitin, we lose this interaction. You see it here compared to here, positive interaction, no interaction. But also in cells that overexpress Aurora A, we have, as we were expecting, an interaction between prohibitin 2 and LC3 itself. So if this data look too complex, you will find a cartoon here indicating you that Aurora A is forming a tripartite complex with prohibitin 2 and LC3, and this complex is taking place only at mitochondria. And again, this is not just cosmetic. What's the purpose of this complex? Well, actually, we started to call these mitochondria super mitochondria. Why? Because actually we provided evidence that if you build this tripartite complex, this is a way for the cell, driven by Aurora A, to degrade a portion of the mitochondrial network and select only the mitochondria that produce high ATP levels. So it's a way, an active way that Aurora A has to degrade the dysfunctional mitochondria and keep only the ones that are super duper good. And why do we say so? Because not only if we have a formation of the tripartite complex, we have high ATP levels, but if we block it using, for instance, pharmacological approaches or mutants, if we block it, not only we restore the mass, but we also restore normal ATP levels. So these super mitochondria are indeed important in Aurora A pathogenesis in cancer, or they seem to be relevant at least. But we're still also microscopy people, so we're not only cell biologists, we are also microscopy people. And I was really frustrated because actually, okay, we depicted a role for the tripartite complex, but what we could see were the entry point, so here, mitochondria were still there in the cell, and we could only detect the endpoint here. So mitochondria at one point, no mitochondria at a later time point. And we couldn't follow the degradation of mitochondria in real time and with a robust tool. The tools that we had in the field were actually not that, were not performing very well, at least not in this paradigm. And so if you're in the microscopy field, you might appreciate the fact that, okay, you don't have a tool, you want a better tool, you better build it. But of course, it also means that you're back to square number one, and you have to start again by validating a new tool. And so this was a project carried out by Begum, a PhD student in the team, and now a postdoc, who actually started to dissect the molecular signaling of the autophagy pathway. So I told you that LC3 is a key player in it, but unfortunately for Bigium that had to deal with this back then, it also has a very complicated signaling cascade. Everything starts right after the production of LC3, and as soon as it's translated, actually it's taken care of by ATG4, which acts as a molecular scissor, and which recognizes this residues and cut this residue here, this glycine, and cuts a portion of LC3. And then this is called priming, so it's a real autophagy initiation step, but then LC3 is undergoing a wide series of intermediate processing steps. And this is the market list. I don't expect you to remember any of this unless you are in the autophagy field and you're interested in this. But you may be interested in knowing that LC3 is literally taken care of by intermediate ATG proteins that actually take it and carry it physically at the interface with the phagophore vesicle, so this vesicle that degrades the material. And at this point, other ATG proteins will conjugate, so will attach a molecule of lipid to LC3 that will allow for the integration of LC3 itself on the phagophore membrane. So you're sticking LC3 on your membrane. And then the vesicle grows and grows and grows until it closes up. And this is a go sign. Autophagy at this point is turned on. And of course, whenever you say that you can turn something on, it means also that you can turn something off. So it's sufficient to remove the lipid here to detach LC3, which is freed in the cytosol, and then the pathway can begin until the source of stress is solved and degradation is no longer needed. Why am I telling you this story? Well, because in the field, and this is part of the reason why the tools we had back then were not sufficiently robust or informative, is that autophagy initiation was back then currently was undetectable, meaning that the great majority of tools were reporting only on this step, and people in the autophagy field use this conjugation step to infer whether autophagy is on or off. But actually, everything that comes before is non-detectable. So if you want to have a more sensitive tool, we reason that we should have designed a sensor to detect autophagy initiation. And again, we went back to FRET, and we built, so we basically flanked LC3 itself with a donor-acceptor pair here. And the FRET readout allows us to say, okay, autophagy is turned off. And wherever autophagy is initiated, remember this ATG4 partner that, remember the scissors, actually it's cleaving off a part of LC3. So the donor and acceptor moieties are actually teared apart, and no FRET is longer detected. And then autophagy can still go on, but we still have a way to sense its progression, because our LC3 fused with the donor is a regular LC3-based construct that sticks to the phagophore membrane, and so we can quantify with a regular confocal microscopy readout the abundance, for instance, of LC3 positive vesicles and infer on the status of autophagy. And for this sensor, and you see me coming, it relies on the improvements and the knowledge that we obtained from the Aurora sensor, because we benefited from aquamarine and TD-LAN YFP, which are a particularly convenient pair of donor-acceptor pair. And why are they convenient in this context? Is because they are totally pH resistant. So they resist to acidic pH. So in theory, this allows us to follow the pathway really from the beginning until the incorporation of the vesicle and its fusion into the lysosomes. So first of all, this was the design, right? So you need a proof of concept that what you design is actually happening. And first, we wanted to understand whether the sensor responds to an ATG4 dependent cleavage. So remember the scissors here. So we compare the wild type sensor, where this residue here is accessible to ATG4. We compared it to a G120A sensor, where by replacing this glycine with an alanine, you basically block the action of ATG4. ATG4 does not longer recognize LC3, and LC3 cannot be cleaved. And this was a go, no go step for us. And you can appreciate here in the images here concerning the wild type and the G120A sensor that we have a dichotomic behavior. Here with the wild type sensor, we basically detect no FRET. Here you have the fluorescence and the delta lifetime cartography. So basically all these pixels correspond to no FRET values. They have a lifetime which is similar or almost identical to the donor. Whereas wherever you have a mutated biosensor here, you see a very significant FRET. And here I would like to drive your attention on the delta lifetime differences. We go from, of course, no variations or very limited variations here with the wild type to 500 picoseconds of delta lifetime difference for the mutant. And here we were taking, so this difference is really much more significant than the Aurora A, for instance, where at maximum we could have 250 picoseconds. But also here we were performing our analysis by calculating the mean delta lifetime, meaning that we get one value of mean delta lifetime throughout the entire cell area. So the entire cell area is one value. And with this type of conditions, it was particularly sufficient, actually, I would say, because it allowed us to determine that the FRET, first of all, here, depends on the presence of this glycine or not, which is targeted by ATG4. If you remove it, you start to see a lot of FRET. And of course, we wanted to take the question the other way around. So not acting on LC3, but acting on the enzyme. And of course, we employed ATG4 siRNAs to answer this question. So here you will always see the mutated version. In the cartography, you see that these huge delta lifetime differences are maintained. You see them here in the pseudocolor scale, where purple means FRET, and you can see it here showcased in the graph. No matter what we do to the mutant, we see this huge delta lifetime difference occurring. But we were actually very disappointed to see that if we look at the wild type sensor in ATG4 siRNA condition, actually we couldn't see any significant difference compared to the control ones. We could see some, but it was actually never or almost never significant. And actually, our frustration when looking at these data led us to ask us a further question. We started to reason that maybe mean delta lifetime fails where pixels don't, meaning that maybe by using just a mean delta lifetime throughout the entire cell area was limiting us in extracting pertinent information still occurring in the cell. And so we implemented a more sensitive type of analysis, which consists, first of all, of what we call a histogram analysis. 5 minutes? No, it's over? Oh, sorry. Okay. I can finish. Okay, thank you. And you will see here that at the end of the tail, we have a shift in the two curves from control to ATG4 siRNA, and you have pixels in the ATG4 siRNA that actually FRET as much as the mutants. So apparently, I'm running out of time. So I'm not going to be telling you this entire story and how this actually is pertinent to uncover new ways of regulation of autophagy during the cell cycle, but I will be happy to discuss with you further should you be interested. And so the original question, and I promise I'm done, the original question in understanding whether this sensor is pertinent for array-dependent mitophagy, well, we still don't have an answer to this question, and maybe next time that we discuss together in Mifobio, we will finally have an answer to this question, but the experiments will be conducted soon. So, in conclusion, I hope I convinced you that FRET sensors are indeed a nice tool to detect the roles of Aurora A and potentially autophagy within cells at mitochondria, that we can image complexes made of different proteins at mitochondria, and that we can implement both broad and sensitive analysis to extract more subtle spatial and temporal information. And so if you want to know more, come and see our workshops. So there is a workshop animated by Claire, PhD student in the team, that is understanding protein colocalizations and mitochondria using STORM. So this is a new line of research for us. And with Xavier Pinson, engineer on the macroscopy facility in Rennes, we're trying to implement methods to both extract the super resolution information and the FRET information. And this afternoon, there is also a roundtable on biosensing for organelle imagings that is animated by Sandrine Pouvreau and myself on new developments and challenges in this topic. And with this, I thank you for your attention. This is our team in Rennes. Ouch. I'm particularly grateful to collaborators that were instrumental in detecting the roles of Aurora A at mitochondria, such as Béatrice Durel, Charles Kevran, Marie, of course, who is here in the audience, that provided us with TD-LAN YFP, and Arnaud Gauthier, who actually engineered hybrid versions of the Aurora A sensor relying on the fast tag. Today I presented you data from Bigium. I'd like to thank the sponsors, of course, and you for your attention. And I'm happy to take questions.
Thanks, Giulia, for nice, beautiful, and clear, interesting talk. Two rapid questions. First, have you tried to see something about P62 on this process? And there exists a mutant where you have, for example, Aurora A, mainly expressed in cytosolic level that reverse the level expression. So there are two unrelated questions.
Yes, okay. So I'm starting with the second one. So yes, we tried mutants that only retain their cytosolic activities.
Yes.
They're really not very nice for the cell. I mean, the cell doesn't like to have cytosolic or only Aurora A. So the mitochondrial pool is really important. And concerning the P62, I guess it's more for the LC3 sensor part. So we are wondering if these huge clusters of FRET that we see in the cells are P62 related. We still don't have an answer for that, but this is indeed on our to-do list.
And then the rest of my question at your round table.
Of course.